dear students, welcome to STUDYHIGH.Pk and subscribe, keep in contact for more video and update. Today I am going to start a new chapter heard lecture 1, medical physiology. First of all, I want to inform you what are the important concepts or topics that I am going to discuss with you in this lecture. The first, the most basic and the most important physiology of cardiac muscles. Second, generation of action potential in cardiac muscles, physiology of calcium ions, T tubules, and different type of ions in generation of action potential, and their transfer from one cell of the heart muscle to other cells. These are the some very important topics that we will discuss in this lecture. So let's start our first topic, physiology of cardiac muscles. So what are the important concepts? C1, C2, C3 are the three important concepts that you will understand after completion of my discussion on physiology of cardiac muscle, like major muscles of heart and their difference from skeletal muscles, cardiac syncytium and role of intercalated discs in cardiac muscles, significance of fibrous tissue in separation of heart syncytium. So these are the three important topics or important concepts that you will understand in this topic. Then we will start our next topic of action potential generation and transfer in cardiac muscles. So let's start our first topic, physiology of cardiac muscles. First of all, I want to inform you our heart consists of three important type of muscles: arterial muscles, ventricular muscles, and conducting or excitable muscles of the heart. Arterial muscles that involve in formation of arteria, ventricular muscles that involve in formation of right and left ventricles, and third type of muscles are those whose force of contraction is almost negligible, and their role is transport of action potential from SA node or pacemaker to arterial muscles and then through AV node to ventricular muscles, and include different type of Purkinje fibers as well. So these are the three important types of muscles. So to understand the role of cardiac syncytium and intercalated discs, you must understand how action potential is generated in a normal cell. Just consider this is a normal cell of cardiac muscle. When action potential is generated in this cell, what will be the situation of this cell? If action potential is generated in normal conditions, in resting condition, the inside of the cell is negative and outside is positive because a large number of sodium ions present outside the cell as compared to the inside, and large number of organic protein acid present inside. This is a resting membrane potential. The resting membrane potential of this cell is minus 90 millivolt, which is correct for most of the ventricular muscles but not for skeletal muscles. So this is the resting membrane potential of this cell, minus 90 milliwatt. When action potential is generated, or you have to generate or you want to generate action potential in this cell, what you want to have to do? First you have to decrease this potential to at least minus 70 milliwatt. So why you have to decrease this potential to minus 70 milliwatt? Decrease, not decrease but increase. You have to make it less negative inside as compared to outside. So as a result, for generation of action potential, you have to decrease or make this inside less negative to at least minus 70 milliwatt action potential, because minus 70 milliwatt action potential is the threshold potential for opening of voltage gated sodium channels that are present in the cell membrane. Like this is a voltage gated sodium channel that is present in the cell membrane. When the inside of this cell, due to arrival of action potential, becomes less negative and it is to minus 70 milliwatt, this sodium channel opens, and when this sodium channel opens, a large amount of sodium that is present outside or in extracellular matrix of this cell start moving towards the inside. When this sodium start moving towards the inside, then a complete depolarization of this cell will occur, and this potential will reaches to approximately plus 20 in heart muscles. And you will say that you have completed your course of generation of action potential, and inside has become plus 20 or positive. When action potential is generated, then the inside of the cell will be positive and outside of the cell will be negative. Why inside of the cell become positive? Because a large number of sodium ions has been transported from the extracellular matrix into the cell. Why sodium has been transported? Because we have converted or we have made this inside of this cell less negative to approximately minus 70. So when the inside of any cell, inside potential or membrane potential of any cell becomes or reaches to minus 70 millivolt, sodium channels open. And when sodium channels open, a large amount of sodium from the extracellular matrix start moving towards the inside, and the membrane become more negative outside and positive inside. Now I am going to show you how intercalary discs involved in transport of nerve impulses or action potential from one cell to other cell. Just imagine this is a cell and it is connected to the other cell of human heart or other arterial cell. It is connected through the intercalary disc that has a large number of gap junctions, desmosomes, connexons, which you will read in histology. So what is the function of these gap junctions? It can transport the positive ions or can exchange a different type of ions between these two channels, between these two cells. When action potential is generated in this cell and its inside become positive, what will happen? A large number of positive ions can move from this cell to this cell who is in resting condition through the gap junctions made or present in intercalary discs. So what happened to the action potential of this cell whose resting membrane potential is minus 90 millivolt? When a large number of positive channels from positive ions from this cell incorporated into this cell, its potential, its membrane potential or resting membrane potential will be disturbed. As a result, its inside become less negative, and when this cell inside membrane potential becomes or reaches to the threshold minus 70 millivolt, the sodium channels present in this cell will also open. When sodium channels in this of sodium water-gated sodium ions or channels open, a large number of positive sodium ions that are present in the extracellular matrix of this cell will start moving inside. When a large number of sodium ions move towards the inside of this cell, this will be also depolarized. Action potential has been transferred from this cell to this cell, and its membrane potential will also reach to plus 20. And this is further connected to the other cell of cardiac muscle through the intercalary disc. And in the same way, the action potential can be transported to all the cells that are connected with the intercalary disc. So now you can understand how cardiac symptoms helpful in transport of impulses from one part of the heart to other part. Our heart consists of two cardiac systems, arterial cardiac system and ventricular cardiac system. So when one cell of the arterial system depolarized or action potential is generated in one cell of arterial muscle, it will be transported to all the cells of arteria that are connected to the intercalary disc. So as a result, all the arterial muscle cells will be contract simultaneously for efficient pumping of the blood from arteria to the ventricles. So because of the system, it does not happen that one cell is contracting and other cell of artery are relaxing. So because of this intercalary disc, impulses transport to all cells of the artery at the same time. Because of intercalary discs, action potential can be transported from one cell to another cell with a large speed with maximum velocity. As a result, all the cardiac muscles, arterial muscles contract with coordination and involved in pumping of blood with efficiency, with full efficiency. Now, in a similar way, like arterial muscles or ventricular muscles are connected to each other through intercalary discs. As a result, when through AV node, one muscle or action potential when generated in one muscle of ventricle, it will be transported to all the muscle cells of the ventricles. As a result, all ventricular muscle will contract with coordination, will simultaneously contract and transport blood to lungs through pulmonary circulation and to other body parts through systematic circulation. So this is the importance of cardiac syncytium. And arterial muscles, arterial syncytium is separated from the ventricular syncytium through a fibrous tissue. Why this separation occurs? Like this is the arterial syncytium, it is separated from the ventricular syncytium through a fibrous tissue. This is a connective tissue that does not allow to transport impulses from arterial muscle cells to the ventriculars, so that arteria must contract before ventricles contract. If this impulse, if there is absence of fibrous tissue, what will happen? All the muscles of heart, arteria, ventricle contract simultaneously. As a result, the heart could not be transport blood with full efficiency. So, because of fibrous tissue, first ventricles contract and transport blood to ventricle. When artery are contracting, ventricles are relaxing. So, when artery contract, ventriculars are relaxing, and as a result, when artery contraction is completed, ventricles contract and transport blood to the lungs and the systematic circulation. Because of fibrous tissue, this is an insulator. It does not allow the impulses or action potential to transport directly from arterial muscle into ventricular muscle. Only a single pathway through which the impulses can be transported from arteria to ventricle is the fibrous tissue or is the AV node or Purkinje fiber that are present in this hair. So only through AV node impulses or action potentials can be transported from arteria to ventricles. So now we have discussed what are the important concepts I have discussed with you. First, I inform you about the major muscles of heart, arterial muscles, ventricular muscles, and their difference from skeletal muscles. Skeletal muscles do not contain intercalary discs. As a result, they are not connected with through gap junctions, whereas cardiac muscles contain intercalary discs and consist of two syncytium. As a result, if action potential is generated in one muscle cell of arteria, it will be transported to all the muscle cells of arteria. Similarly, if action potential is generated in one muscle cell of ventricle, it will be transported to all the muscle cells of ventriculars. So intercalary discs involve in transport of action potential from one cell to other cell. It provides a very low resistance. Significance of fibrous tissue in separation of heart syncytiums. So heart syncytium involved in contraction of arteria. Arteria should be fully contracted and should be transported all the blood they contain into ventricles. And when arterial syncytium is completed, then ventricle should contract. As a result, heart should pump blood to all body parts with full efficiency. So we have discussed the physiology of cardiac muscle. Now we are going to discuss the next topic of our lecture, action potentials in cardiac muscles. Three important concepts that you will be understand after completion of my discussion on this topic: difference of action potentials in cardiac muscles and skeletal muscles, why duration of ventricular contraction is 15 times larger than skeletal muscles, different phases of cardiac muscles. These are the three important concepts that you will be understand after completion of my discussion on this topic. So let's start discussion on this topic. First of all, you must know, and I have already explained how action potential is generated. So now we are going to discuss in detail. This is a cell membrane of cardiac cell whose inside is negative in resting condition and whose voltage or resting membrane potential is minus 90 millivolt and whose outside is positive in resting membrane conditions. When action potential has been generated or you want to generate action potential in this cell, first you have to enter or action potential and other cell or in from SC node will transport some sodium ions through gap junctions or intercalated discs into this cell cytoplasm. As a result, its resting membrane potential has been changed from minus 90 millivolt to minus 70 millivolt, which is a threshold potential for this sodium channel. When threshold potential for opening of the sodium channel has been reached, a large number of sodium ions that are present in the outside or in extracellular matrix of this muscle cell will start moving in this cell through this sodium channel. So sodium influx, due to the large number of sodium influx, its inside voltage or membrane potential will become positive, and inside become positive and outside will become negative, and its membrane potential during the action potential will be plus 20 because of this sodium or influx of the sodium ions through the resting membrane potential. Resting membrane potential is minus 80. Just think minus 85 here. This is a minus 85 milliwatt, which is the resting membrane potential for a cardiac muscle. This is milliwatt. So what happened due to the opening of sodium ions, sodium channel, this potential has been reached to plus 20. This is a figure of plus 20. So influx of sodium ions into the cell causes the rise of plus 20 milliwatt. This is the plus 20 milliwatt. So what happened next? A large amount of sodium has been entered. This is the phase 0, phase of rapid depolarization. The membrane that is in polarized form initially has been gone into depolarized form because of a large number of sodium from the outside or from extracellular matrix has been moved into the cell. Now in next step, this is the phase 0, which we call phase of rapid depolarization. Now we are going to discuss what will happen in next cell or in next step. In the next step, there are two types of channels. One is rapid or fast potassium voltage gated channels, and second are the slow potassium channels. You must know about two types of channels that involve in repolarization of cell membrane of cardiac muscles, whereas it also consists of a number of potassium channels that involve in controlling the action potential and state of cardiac muscle cells. First, fast potassium voltage gated channels. These open very quickly and also closes with the same speed with which they opens. These are the fast potassium channels. And second are the slow potassium channels that involve in causing or controlling the repolarization of cell membrane of cardiac muscles. So I am going to explain you how these two muscles, these two potassium channels are involved in during different phases of cardiac muscle cycle channels. So due to influx of sodium, the membrane potential has been reached to plus 20, is now reducing initial repolarization because of which channels? Because of fast potassium channels. But these fast potassium channels, after initial a little repolarization, closes simultaneously with the same speed with which they open. The next step that happens in cardiac muscles is the plateau caused by the potassium channels. This plateau is because of the potassium channels. When the rapid or fast potassium channels closes here, calcium channels open. Which calcium channels? In cell membrane of cardiac muscles, there are large numbers of L-type calcium channels. L-type mean long lasting. They are the slow calcium channels. So long lasting calcium channels or slow calcium channels open. What will happen as a result? A large amount of calcium will also start moving towards the inside of the cell. As a result, there is a plateau. So this plateau does not happen in skeletal muscles. Because of this plateau or opening of calcium channels, membrane remain depolarized for 0.2 to little slow L-type calcium channels involved in causing this plateau. So as a result, what will happen? A large number of potassium or calcium also enter to inside. Second, these calcium channels temporarily also, so it also decreases the permeability of potassium ions to move inside when calcium is being transported to the inside of the cell. As a result, the membrane rise for some time. After calcium ions start closing, or L-type calcium ions that are present in cell membrane of cardiac muscles, when closes, then slow potassium channels open that will cause the rapid repolarization. So these are the different phases. Phase 1, phase 0 include the rapid depolarization by sodium channels due to influx of sodium, potential reaches to plus 20 milliwatt. Phase 1, this is a phase 1, initial repolarization because of fast potassium voltage gated channels. Then there is a plateau phase, phase 2, which is a plateau phase because of calcium channels or L-type calcium channels present in cell membrane of cardiac muscles. And here is a phase 3, which is because of opening of slow potassium channels that involve in repolarization of cardiac muscle cell, showing the different types of steps that are occurring in cardiac muscles. First, 0 rapid depolarization because of sodium channels and action potential or membrane potential in millivolts has been increased from minus 85 to plus 20 millivolt. Then initial depolarization stage 1. In this, because of opening of rapid potassium channels, and there is a plateau phase, you can see, in which calcium channels, L-type calcium channels opens and cause the movement of calcium ions into the cell membrane and increases the time of contraction of the cardiac muscles. This is only a characteristic of cardiac muscles. After this, a rapid opening of slow potassium channels. So these are the three important phases. In phase 4, what will happen in phase 4? Phase 4, the original membrane potential or resting membrane potential has been restored by using a different type of sodium potassium ATPase pump. Similarly, calcium ATPase pump present in sarcoplasmic reticulum and different types of calcium sodium exchangers etc. During phase 0, a large number of sodium has been transported to the inside, which causes the increase in membrane potential from minus 85 to plus 20. In the next phase, there is a initial repolarization, a little bulge as a result of because opening and at the same time closing of some fast voltage gated potassium channels. And after that, a plateau phase occurs during which calcium ion is rushed into the cell because of opening of L-type calcium channels. And after that, there is a rapid depolarization because of opening of slow potassium channels. So these are the different types of ion concentration that affected during the. So in this topic we have discussed or I explained you how action potential is generated in cardiac muscles and how it is different from skeletal muscle, because cardiac muscle consists of L-type calcium channels which the skeletal muscle cells lack. As a result, there is a plateau in cardiac muscles after depolarization, whereas there is no plateau in skeletal muscles. Skeletal muscle simply consists of a rapid depolarization and after depolarization there is a depolarization because of potassium ions. So there is a no plateau in skeletal muscles, whereas cardiac muscles consist of a plateau. They remain depolarized for some longer time as compared to skeletal muscles. So because of this reason, ventricular contraction, duration of ventricular contraction is 15 times larger than skeletal muscles. And I have also explained you different phases of action potentials of cardiac muscles: rapid depolarization, initial depolarization, plateau phase, and rapid depolarization. These are the different phases of action potential in cardiac muscles. Now we are moving towards our next lecture or next topic, that is velocity of impulses in cardiac muscle and refractory period. Now we are going to discuss the velocity conduction in cardiac muscles and other types of different units that conduct action potential from one part of the body to other part. In heart, the conduction velocity of arterial muscles or fibers that conduct impulses from SA node to AV node is between 0.3 to 0.5 second. 0.3. Now we are going to discuss the velocity conduction, the conduction velocity of action potential in cardiac muscles and other type of nerve fibers and skeletal muscles, body units that conduct action potential from one part of the body to other part. Like in heart muscles, the velocity or conduction velocity of action potential is approximately 0.3 to 0.5 meter per second. Whereas in nerve fibers, large nerve fibers that conduct impulses from different body parts to the brain and spinal cord, their conduction velocity is approximately 250 times larger as compared to the conduction velocity of heart impulses. Because of their functions, the difference in function, their coordination. In heart, we require a coordination between atria and ventricle, which demands that the impulses should be travel slowly as compared to the nerve fibers, so that atria should be contracted before ventricular contraction. So that's why heart muscles, the conduction velocity of fibers of heart is less as compared to skeletal muscles and other type of nerve fibers. In skeletal muscles, the conduction velocity is approximately 10 times that of heart muscles. Now we are going to discuss the most important refractory period. What is absolute refractory period and relative refractory period? If from the phase 0 to the middle of phase 3, this period is called absolute refractory period. What is refractory period? The period during which a cardiac muscle or muscle cells could not be re-excited, who are already in excited or contracted or depolarized form. So this is the period during which a cardiac stimulus cannot excite an already excited area of heart, no matter how much strong stimulus or action potential you are providing. So this period is called absolute refractory period. And why this happens? Because a large number of sodium during action potential has been entered inside the cell. The incorporation or influx of large number of sodium causes the or inactivated. It causes the inactivation of sodium voltage gated sodium channels. So voltage gated sodium channels are inactivated. That's why no action potential cannot be generated in this muscle cell who is already in depolarized form. When after half of the rapid depolarization, many of the potassium ions that are present inside through different channels will be opening of voltage gated sodium channels. As a result, if a nerve impulse, if a strong stimulus or action potential provides during this period, this muscle will be contracted. But here is a refractory period during which no cardiac muscle can be re-excited. no matter how much strong stimulus you are providing. This is a period of relative refractory period during which a strong stimulus or action potential can re-excite an already excited area of cardiac muscles or muscle cells who is being recovering from the action potential. If a strong stimulus causes a contraction, heart muscles who are recovering from the action potential will be contracted, but the force of contraction will be very low as compared to the normal. So this is the reason of early premature contraction and later premature contraction depends at what stage of the or at what conditions of cardiac muscle cell you are providing. So first of all, you must know the composition of T tubules, how they can affect or transport the calcium ions between the cell and extracellular matrix. First, this is a T tubule. As you can see, this is a T tubule. What is the basic function of this T tubule? When action potential is coming from this direction, this T tubule transfers the action potential to the interior of the cell. So this is different type of organs, sarcoplasmic reticulum, etc., and myofibrils, actin, and myosin filament. So this is our cardiac cell covered by sarcolemma, and this is a T tubule. The configuration and composition of T tubule in cardiac muscle is different as from skeletal muscles. This is 25 times larger in volume as compared to skeletal muscle, and it consists of a different type of disaccharide, negatively charged disaccharide polysaccharides, polymer of negatively charged disaccharides called GAGs, glycose aminoglycans. If you have read in your chapter carbohydrate, glycose aminoglycans that consist of galactidonic acid, hyaluronic acid, different type of keratin sulfate, these things. So its wall consists of a large number of GAGs that are negatively charged and that can store calcium ions. So as a result, a large number of calcium has been stored in the wall of T tubules because it contains a large number of mucopolysaccharide. So what happens when an action potential arrives on the surface? The first function of T tubule is to transfer the action potential from the surface to the core of cardiac cell. As you can see, the action potential coming from this and enters to this junction because of the first action potential from our surface to the core of the cell. Then after transfer, what happen? What are the different processes that are occurring? There are L-type calcium channels present. L-type mean long lasting, present in the membrane. So first, what happens when action potential came? The calcium that is present in its cell membrane bound with the mucopolysaccharides, GAGs, it start diffusing towards the inside of the cardiac cell. So first step, when action potential arrives in the T tubule, the calcium that is being stored in the T tubule start diffusing into the cardiac cell. The diffusion of this calcium causes the opening of this L-type calcium channel. This is an L-type calcium channel. So this opening of this L-type calcium channel causes the influx of large amount of calcium that is present in extracellular matrix. So the opening of this potassium calcium L-type calcium channel causes the influx of a large amount of calcium in the extracellular matrix to move into the cell. This calcium moves in. So from activation of L-type calcium channel, a large amount of calcium that is present in extracellular matrix of the cardiac muscle cells start moving towards the inside of the cardiac cell. So when a large number of calcium has been incorporated in the inside of the cell, it causes two important things. So what are the important functions it can provide? So this calcium that is here incorporated into the cardiac muscle cell first causes the activation of ryanodine receptor cells. What are the ryanodine receptor cells? Just imagine here is a sarcoplasmic reticulum that is present in our muscle cell. It consists of a ryanodine receptor cells. Ryanodine receptor cells is activated because of a large amount of calcium that enters into the cytoplasm of the cardiac cell. So this ryanodine receptor cell on activation release the calcium present in sarcoplasmic reticulum into the systole. And second important function that they perform, when a large amount of calcium incorporated into the cardiac cell, it causes the inactivation of L-type calcium channel. On inactivation of L-type calcium channel, further calcium influx of calcium from the extracellular matrix is stopped, and the calcium that is present in sarcoplasmic reticulum start moving outside into the cytoplasm of this sarcoplasmic reticulum or cytoplasm of our cardiac cell. Then further process that occur here, this calcium that is moving from the sarcoplasmic reticulum to the outside, and the calcium that has already entered in the cytoplasm of the cardiac cell from the extracellular matrix through tubule, through L-type calcium channel, and through mucopolysaccharide is then moved towards the contractile unit. What is contractile unit? Actin and myosin filaments, like you have read in your FSC intermediate level and in your previous chapters. So the contractile unit, actin and myosin, as you know that the thin filament consists of a troponin, tropomyosin. It will bind to troponin, then causes the bridge formation between the actin and myosin and the contraction of muscle. So now I am repeating the whole process that is happening here. What is basically happening during different types of impulses that arrive here. First, a nerve impulse arrives on the surface of the cardiac muscle cell. T-tubule causes this impulse to enter to the cord of cardiac muscle. On arrival of this action potential, the calcium that is bonded with the mucopolysaccharide of T-tubule start diffusing into the cardiac muscle cell. On diffusion, what they cause? They cause two things. First, this calcium that is diffuses into the cell causes the activation of L-type calcium channel. L-type calcium channel causes the influx of a large amount of calcium that is present in extracellular matrix to flow into the cardiac cell, which on entering into our cardiac muscle cell causes two things. First, it inactivates the L-type calcium channel. It inactivates it, and second important function that this calcium, a large amount of calcium that has entered in our cytoplasm of cardiac cell perform is the activation of ryanodine receptor cell present on the sarcoplasmic reticulum. So on activation of ryanodine receptor cell present in the cell membrane of sarcoplasmic reticulum, a large amount of calcium present in or stored in sarcoplasmic reticulum start moving outward into the cytoplasm of cardiac muscle cell. This calcium then moves to the contractile unit actin and myosin and cause the contraction of cardiac muscles in the similar way as they do in skeletal muscle. So this is the basic process that happens. So what happens when the muscle is contracted? No, our cardiac muscle or skeletal muscle is going to relax. The calcium that bonds with troponin is released. A large amount of calcium that enters here stored in the cytoplasm is moving in backward direction. First, what happens? This is sarcoplasmic reticulum. It contains another calcium ATPase pump that causes the movement of calcium from the systole back into the sarcoplasmic reticulum. The calcium that has been used by the contractile unit and the extra calcium is then moving backward through the calcium potassium ATPase pump into the sarcoplasmic reticulum. So this calcium is moving back. So in first step, through ryanodine receptor cells, calcium is moving out. Now through calcium ATPase pump, it is moving back. Now here is the cell membrane of our cardiac cell. It consists of calcium exchanger that results in moving of sodium into the cell and moving of calcium outside. As a result, what will happen? The calcium will move outside and sodium will move inside. Calcium is moving outward, sodium is moving inward. So this is the sodium and the calcium is moving onward in this direction. So the sodium that got entered through this pump or other processes then move outward in outward direction through the sodium ATPase pump. Now axial potential that arrives through this T-tubule. This axial potential coming through T-tubule causes the movement or diffusion of calcium that present here into the cytoplasm or cytosol of our muscle cell. As a result, what happens? It causes the activation of these L-type calcium channels. On activation of these L-type calcium channels, calcium, a large amount of calcium that is stored in extracellular matrix, start moving towards the inside. On going inside, it causes the activation of ryanodine receptor cells. And on activation, a large amount of calcium can pass through the ryanodine receptor cell into the cytoplasm. These then go into the cytoplasm of then go to the contraction unit. Like you must know that how our calcium involved in contraction of actin and myosin filaments. So they will go to here, and on going here they cause the contraction by bonding to the thin filaments etc. So no a large amount of these muscles are relaxed. Now there is a large amount of calcium that is present here. Now what will happen? So the calcium that is present here is start moving back into the sarcoplasmic reticulum through calcium ATPase pump, and calcium is moving backward, and those calcium that is present in our muscle cell and should be transported to the extracellular matrix, because calcium also enters from extracellular matrix. To maintain the concentration of calcium, the calcium should be moved backward into the extracellular matrix as well. So the calcium also moves outside through the calcium sodium exchanger pump that is present in sarcolemma of cardiac muscle cell. And the sodium that got entered during the whole process is moved outside through the sodium ATPase pump. So this is the basic process that is occurring in our human heart muscles or cardiac muscles. So the concentration of calcium in extracellular matrix is very important for cardiac muscles, because the sarcoplasmic reticulum that involved in secretion of calcium or releasing of calcium ions is very thin in heart muscles. That's why our for contraction or continuous contraction, our heart muscle also depends on the large amount of calcium present in extracellular matrix. So if a diet of anything, any injection that increases the calcium concentration can increase the force of contraction. Similarly, if calcium is decreased, heart contraction will be slower. So this is a clinical related. You must know that what is the effect of calcium, high calcium concentration in extracellular matrix and low calcium concentration, and these things we will discuss in our coming lectures. So this is our first lecture. Hope you understand how different processes are occurring in our human body, physiology of cardiac muscles. Then we will discuss physiology of action potential in cardiac muscles, how they differ, different type of phases, why ventricular contraction duration is 15 times greater than skeletal muscles. We have also discussed the velocity of impulses and refractory period. What is absolute refractory period? Its duration, at what phase it starts and at what phase it ends. It starts at from phase 0 and ends between the middle of the phase 3. And why it occurs? Because of inactivation of sodium channels. And physiology of calcium ions and tubules we have discussed. Hope you best of luck. Like, subscribe, share. Thank you. Allah Hafiz.